So stable carbon isotopes, this is a set of equations which explains how the two main variables are defined for stable carbon isotopes. So there's little delta right here, which ends up calculating for plants, for trees, as a negative number. And then there's big delta here, which ends up calculating for our cases as a positive number. So for little delta, more negative equals more discrimination against 13C. With big delta, higher positive numbers equals more discrimination. Okay, so that's a key thing to remember as you're looking at these variables is those facts. So I'm going to erase those so those aren't too distracting. So let me talk about how these are calculated. Little delta is calculated relative to the carbon 13 to carbon 12 ratio in a standard. So that's this R standard. For when these were developed, and this technology was developed for geology, I believe. It wasn't developed for use in plant physiology. But when it was developed for geology, they used a reference for C12 to C13. The geological reference they used was belemnite. It's a mineral, and they used a particular structure that was located in PD, North Carolina. So they use PD belemnite. And this isn't important to remember, but sometimes you'll see relative to PDB. PD belemnite is what they're talking about. So that's this R standard. So that R standard is the molar ratio of carbon 13 to carbon 12 in that PD belemnite standard. So when this was developed, they used actual chunks of rock from this big rock in PD, North Carolina. Now they have manufactured standards that have that same C13 to C12 ratio. And then we look at the ratio of carbon 13 to carbon 12 in the sample that we're measuring. So for instance, that might just be dried and ground leaf tissue. And that's measured in a mass spec, and I'll show you in a minute how that's measured. So we get this R value, and it's expressed relative to that standard. So everything's expressed relative to a standard. And so what that means is that relative to PD belemnite, the C13 to C12 ratios are all negative relative to PD belemnite. So this little delta ratio for leaf tissue, for instance, is always some negative number. Okay, so that's one little delta measurement, and we'll see some examples of these in a minute. That's always negative. There's another measurement, big delta, which basically expresses this little delta here. So that's delta plant relative to delta of the atmosphere. So that big delta is just expressed, it's the plant relative to atmosphere, and it's a positive number. So bigger positive numbers for this means more discrimination against C13. Larger negative numbers for this little delta means more discrimination against C13. Farquhar et al. In this paper, if you look this paper up, they do the theoretical work to show that big delta is related in this way to the CI to CA ratio. So CI to CA ratio basically is controlled by B and A, where those represent the relative discrimination due to stomatal resistance and the relative discrimination due to Rubisco. So all of that theoretical work was worked out by Farquhar. So if you want to dig down into the weeds, that's a good one to look at to understand the mechanics of all of these values. But suffice it to say that these delta numbers are just a time-integrated measurement of the relative photosynthesis to conductance ratio of plants. And that's where the value of these comes from. Now, one problem with this is that when we get a stable carbon isotope ratio value, either little delta or big delta, that just tells us a ratio of photosynthesis to conductance. So if we see that delta is different in two different situations, we don't know if delta is different because photosynthesis changed. So again, remember, this is the ratio of photosynthesis to stomatal conductance. We don't know if delta is different, if it's different because photosynthesis changed, or if it's different because conductance changed, or if it's different because they both changed. So in order to wrestle with that, people sometimes use the oxygen isotope ratio. So they can look at stable isotopes of oxygen and the relative amounts of O18 in water in the leaves or in tissues. And that's affected by a couple of things. So this diagram sort of nicely summarizes discrimination against C13 in leaves, which is basically due to the ratio of Ci to Ca, and how that influences stable carbon isotopes in trees. And then oxygen isotope ratios. That's influenced by both the relative amounts of O18 in water taken up through the soil, as well as the rate of transpiration in leaves. So O18 is discriminated against. It doesn't evaporate as readily as other isotopes of oxygen. So rates of transpiration influence the relative amount of O18 in water in leaves. And we can measure that and basically get an estimate of the GS part of that ratio. So if we measure both stable carbon isotopes and stable oxygen isotopes in leaves, then that gives us an idea of whether that change in carbon isotope ratio was due to changes in photosynthesis or changes in conductance. So if we measure both, we can then start picking apart what caused that change. And those are some really powerful approaches to understanding long-term changes in physiology due to the environment, due to experimental manipulations, due to genetics, or due to other reasons. Now, stable isotopes in tissues or in water are measured with a mass spectrometer, and that's what this instrument is. This is a mass spectrometer. It's a pretty old picture, so they may look a little differently now, but the principles are the same. In this case, this mass spectrometer is hooked up to an elemental analyzer on the front end, and what you do is you dry, if you're looking at leaf tissue, for instance, you dry the leaf tissue, grind it up and homogenize it real fine, put it in little tin capsules, and put it in this device. That drops a tin capsule into this elemental analyzer. It flash heats it and vaporizes that tissue and turns it into gas. And then that gas flows through columns in the elemental analyzer, and you usually can analyze, for instance, carbon concentration and nitrogen concentration, sometimes other elements. But then you now have a gas stream that flows into the mass spec, and that mass spec can look at the relative molecular weights of the elements in that gas stream. So this is a diagram that shows how that mass spec works. So that gaseous sample is vaporized here in that elemental analyzer. And then those ions are accelerated and go through a really powerful magnet. And that magnetic field deflects the lighter elements, the lighter atoms, more than the heavier atoms. So there's a detector here which can detect strikes by the heavier atoms relative to the number of strikes by the lighter atoms. And the ratio of those strikes, for instance, carbon-13 versus carbon-12, it can tell you in that sample what the relative amount of carbon-13 atoms are relative to carbon-12, or oxygen isotopes, for instance. Really powerful technique that can be used to look at those ratios in that sample, for instance, that you put into the elemental analyzer. And that's how you get the data that we're going to look at. There are many ways that stable isotopes have been used to analyze plant physiology. And again, stable isotopes of different elements are used in many different fields, not just in plant physiology, for instance. They're used in geology, food science, zoology, many fields. Very powerful. At any rate, we're going to focus on plants. This is an example that's really neat. They're looking at stable isotopes in cellulose in tree rings, and they're indicating this is little delta. So what you're seeing is more negative values means more discrimination against carbon-13. And then the white zones are early wood, and the tan zones, this dark zones, are late wood. This is really neat because the way they did this, they had a sample of wood and they had a laser that was vaporizing the wood. So the laser would vaporize the wood, feed that into an analyzer and a mass spec like I just showed, and move along the sample of wood so that they could get, with fine resolution, the stable carbon isotope ratio of that wood within a growth ring. So you can see they're catching very fine variations in stable carbon isotope, which is really interesting. So you can maybe get an idea of what physiologically was going on during the time of formation of early wood and during time of formation of late wood. Now, it's important to remember that, for instance, wood isn't just formed with current photosynthate. There's stored carbohydrates that go into that as well. So it's not necessarily a direct reflection of physiology at the time those cells are laid down. But it's a neat integrated effect. So there's a couple of things you can see. You can see, for instance, during this year, discrimination against C13 was much lower, less negative values. That's likely was during a very dry period, because a dry period would have more stomatal closure and therefore less discrimination against C13 compared to, for instance, a period like this, which is probably really wet when the stomata were much wider open. So really neat ability to discern both over years and also within years variation in physiology. So that's a neat example. Another example, this was looking at genetic variation in stable carbon isotopes of trees in a progeny trial. So these are different, each dot is a different genotype of tree, and those trees are different size. So this is sort of a graph of tree size or tree growth rate versus carbon isotope discrimination. So this is big delta. So this is hard because you have to, sometimes people use big delta, sometimes people use little delta. In this case, we're using big delta. So in this direction, this is greater discrimination against C13. So again, it appears that there's a correlation between that CI-CA ratio, or the degree of discrimination, and tree size. In this case, bigger trees have less discrimination. One way of interpreting this would be that potentially photosynthesis rate is higher in these larger trees, and that is resulting in less discrimination and potentially greater rates of growth. You'd need to do additional measurements, but stable carbon isotopes are a nice way to screen, for instance, large numbers of individuals for physiological traits. This is another example. This was in a study that had 4 different treatments. And those 4 different treatments, there's a key here. There's C, that's control. D is this one, that's through fall diversion. So they had troughs in the understory of the forest that removed about 1/3 of the water coming into the stand and took it off of the plot. So basically that's a drought treatment. F is fertilizer, and then FD is the fertilizer plus through fall diversion combination treatment. And what you can see is that this is water use efficiency. That is basically carbon isotope discrimination. This is derived from carbon isotope discrimination. And you can see that the plots that received through fall diversion, or the drought treatment, had greater water use efficiency, as measured by stable carbon isotope discrimination. You can see they also measured oxygen isotopes. So that tells us that there was decreased delta 18 OC. So that means there was less transpiration in these plots that received the drought treatment. So that tells us that this change in water use efficiency, or change in carbon isotope discrimination, was due primarily to changes in transpiration or stomatal conductance. So this is an example of how you can use those 2 in combination to understand what was changing, in this case, in this manipulative experiment. This is a pretty interesting one. In this study, the authors are looking at little delta 13C, not of tree tissues, but they're looking at delta 13C of CO2 emitted from soil respiration. Okay, so what they're doing here is you have soil with trees growing in it. And this is a little crooked because I'm drawing on this pad. And, you know, these trees are rooted in the soil. And this delta number here is measurements of CO2 being emitted from the soil. So basically what they're measuring partly is carbon isotope discrimination of respiration from the roots. And it's plotted against air relative humidity 3 to 4 days before. Okay, so let's think about this for a minute. You're measuring delta 13C of basically respiration from the roots. And what they're finding is that that is very tightly correlated with relative humidity several days before. And what we're seeing is that there's less discrimination when there was lower humidity 3 or 4 days before, and greater discrimination when there was higher humidity 3 to 4 days before. So what that's telling us is that we might expect there to be, for instance, if we think about discrimination, on days with lower humidity, that causes stomatal closure. And we might expect then less discrimination against C13C. So less negative values of D13C. On days with higher humidity, we might expect the stomata to be wider open and there to be less discrimination or more discrimination against delta 13C. And the fact that the best correlation is 3 to 4 days prior, that tells us that basically the starch or carbohydrates that are being respired by the roots were fixed by the leaves 3 to 4 days prior. So the humidity 3 to 4 days prior determines the delta 13C of carbohydrates that are then retranslocated to the roots and then respired 3 to 4 days later, and we're seeing that signal in that respired CO2. Really neat study, very indirect, but it tells us something in this case about how long it takes for carbon that's fixed in the leaves to move down and be respired by the roots. This graph shows some data of a survey of a couple hundred species of orchids by Katja Silveira with the Smithsonian Tropical Research Institute. And Katja was doing this work because she was interested in understanding the photosynthetic pathways used by different kinds of orchids. Orchids are super diverse. There's hundreds and hundreds of species. So we don't necessarily know anything about the physiology of a lot of these species. So rather than having to do gas exchange on each different species of orchid, she could just collect tissue from all these different species, put it through a mass spec, and look at its stable carbon isotope ratio. And what she found was that there were two peaks in the population she was looking at. There was this peak here, and of course this other peak here, right here. And you could sort of think about this. You could think about, okay, we might expect some of these to be CAM photosynthetic pathways and some of these to be C3 photosynthetic pathways. So which is which? Let's think about which is which. You might even pause it and think about which of these peaks are C3 and which is CAM. So C3, CAM, let's start with CAM. CAM photosynthesis occurs with the stomata closed. So uptake of carbon within the leaf happens during the day when the stomata is closed. So basically, that's that situation in that initial schematic I showed you. If the stomata were completely closed, almost all the C13 would eventually be taken up by rubisco. That means there'd be very low, less discrimination against C13. So what that means is that this set of species of orchids are CAM, as opposed to this set of species are C3, because they're showing discrimination characteristics more commonly associated with C3, where they're fixing carbon with their stomata open, so there's more discrimination against C13. Now, the actual story in the paper is a little bit more complicated than this, but suffice it to say here, for broad surveys of pathways, carbon isotope discrimination is really powerful for that as well. This final one's pretty fun because I like beer. This is basically from the food science area. This shows how we can use carbon isotope discrimination measurements to determine whether beer is made primarily with sugars from C3 plants or C4 plants. So traditionally, beer is made from barley. Barley is a C3 plant. And so you can see they're showing here C3 plants have carbon isotope discriminations within this range, whereas C4 plants have a carbon isotope discrimination in this range. So basically corn would have carbon isotope discrimination in this range, barley carbon isotope discrimination in this range. Okay, what happens then if we measure the carbon isotope discrimination of alcohols in beer? By looking at those values, that can tell us, for instance, whether beer comes predominantly from sugars derived from barley, or whether it has sugars with at least some amount of corn in it. So these beers basically, because they're shifted in this direction, that tells us they were made with some corn sugar added. And what you can see is that USA beers tend more in this direction. basically cheaper beers, they add corn sugar to the beers for in the fermentation stage because it's cheaper. Higher quality beers tend to be almost exclusively barley, and they would have a carbon isotope discrimination signal here. So that's a neat way of detecting, for instance, what kind of plant sugars were used to make that particular kind of beer. Many other examples of application of carbon isotopes in the plant sciences out there, but this sort of gives you a flavor of how they might be used.